Right. And our last and certainly not least presentation today and of the 2020 Joe Sugg Graduate Student Paper Competition is entitled GFP-tagging of Bradyrhizobium strain Lb8 isolated from peanut Arachis hypogaea linnaeus nodules. And our presenter today is Zifan Zhao.
Can everyone hear me?
Yes, we can.
Okay, good. Good morning, everyone. My name is Zifan Zhao. I'm from the University of Florida, and I'm glad to be here to present my research. My research is GFP-tagging of Bradyrhizobium strain Lb8 isolated from peanut nodules. So my research is focusing on the plant and microbe interaction. So here are the outlines of my today's presentation. First, let's get down to some basic points of the symbiosis. So we know that most of the legumes can form the symbiotic relationship with the rhizobium. The rhizobium is a kind of slow-growing, it's a kind of soil-borne bacteria which can fix the nitrogen in the air. And in most of the legumes, the bacteria can get into the plant cells through the root hair, and it forms an infection thread. So this is a kind of intracellular infection pathway. However, in peanut, the bacterium, the Bradyrhizobium, is a kind of slow-growing rhizobium. It can infect the peanut through the crack entry. The crack entry is like the bacteria can get into the plant cells through the cracks, and the cracks are usually created by the emerging of the lateral root. So here in this picture, this is the main root, and this part is the lateral root. So the bacteria can get in at this point. And this is a kind of intercellular infection pathway. So the crack entry is kind of unique because it only happens in 25% of the legumes and is not fully understood. So for the current research, we are lacking the detailed microscopic evidence to show how this process works. And also we are lacking the fluorescent tagged bacteria strain to study this kind of interaction. So my hypothesis is during the crack entry, the Bradyrhizobium infects the peanut through the mid-lamella between the two adjacent axillary root hair cells. And my two objectives are, the first one is construct the GFP-tagged Bradyrhizobium. The second one is trace the infection pathway of crack entry at different time points. So let's talk about the materials and methods. Before we want to label the bacteria, we need to find the right bacteria to be labeled. So previously, we isolated 5 Bradyrhizobium strains from the peanut nodules. And based on the growth curve and also the inoculation test, we finally selected the Lb8 owing to its relatively fast-growing nature and the higher number of nodules. So this is the growth curve. We can see the right line shows the growth curve of the Lb8. In the first weeks, the Lb8 growth is the fastest. And this is the inoculation test. And this gram shows the number of nodules induced by the Lb8. We can see the number of nodules is the highest among all these 5 strains. So after we selected the right bacteria, the next thing is to put the GFP plasmid into it. The GFP plasmid will be modified first and then transferred into the competent E. coli cells, which is called the DH5 alpha. And from the DH5 alpha, the plasmid will be further transferred into the Bradyrhizobium strain Lb8 through the triparental conjugation under the help of the E. coli strain HB101. And after this whole plasmid get transferred into the Lb8 strain, it will be integrated into the bacterial genome to make sure it is a kind of stable, it's stable expressing. And then we will do antibiotic selection and do an infection test to kind of prove this is successful. So this slide shows more details about how the plasmid modification works. The reason why we do the plasmid modification is like before, we didn't insert the plasmid into the genome, and we found like the plasmid was quickly get lost in the bacteria without the integration. And if we want to insert the whole plasmid into the bacterial genome, we have to put a homologous region into the plasmid. So we amplified a 1 kb region from the Lb8, and this region will be inserted into the plasmid to serve as a homologous region. And this region is from a nodulation independent region. So the insertion of the whole plasmid into the genome won't affect the symbiosis ability. And this image shows like how the plasmid looks like before the enzyme digestion and after the enzyme digestion. And this is our 1 kb PCR product amplified from the Bradyrhizobium strain Lb8. And here we can see these are the two restriction enzyme we used to linearize the plasmid. And then the 1 kb product will be integrated into this region, and they form a new plasmid. So after the plasmid after the bacteria has taken the plasmid, we need to do an inoculation test to make sure like finally the nodules are showing the green fluorescence. We first germinated the seeds, and then we marked the infection site, and then we cut the roots using the cryostat, and we observe the slides using the fluorescent microscope and the confocal microscope. So here are the results. These are the image showing like the E. coli strain taking the modified plasmid. We can see the cells are showing the green fluorescence signal. And this is the strain, the E. coli strain HB101 with the conjugated plasmid PK2013. It serves as the helper. And this is our Lb8 strain. And this is the recipient during the triparental conjugation. So after the conjugation, we put the product on the agar plate with two kinds of antibiotics, the chloramphenicol and the tetracycline. The chloramphenicol is from the Lb8 itself, and the tetracycline is from the plasmid. So only the Lb8 having the successful integration of the plasmid can survive on these two antibiotics pressure. And from here, we can see the single colonies are visible 1 week after the plating. And here are the image shows how the GFP-tagged Lb8 looks like under the microscope. And this slide shows how the infection test was done. Like here, we prepared the inoculum first from a single colony of the positive Lb8 strain. And then the peanut seeds was sterilized and germinated. After the seed was germinated, we inoculated the seeds and planted the peanut seeds into the soil. And 2 weeks after the planting, we dig out the plants and check their nodules. So these are the microscopic image taken by the stereo microscope. The upper panel shows all the image under the white light, and the lower panel shows all the image under the GFP filter. We can see this one is an intact nodule. It's showing the green fluorescent. And from here, the nodule was cut into half-half to enable us to take a closer look to the inner part of the nodule. And this is the zoom in part of the nodule harvard. We can see only the inner part of the nodule is showing fluorescent because the bacteria is in the inner part, but the outside is the plant tissue which do not have the fluorescent. And the last image is a negative control. It's inoculated by the original Lb8, and we didn't see any GFP signal from the nodule. And this slide shows how the image looks like from the confocal microscope. From here, we can see the inner part is showing a very strong green fluorescent from the bacteroid, but the outside is from the plant autofluorescent. And these results match with the previous microscopic observation. So my conclusion here is the GFP-tagged Lb8 is stable and successful. And my future steps is to trace the infection pathway in the symbiosis with the GFP strain at different time points, at like 2 days after inoculation, like 4 days after inoculation, and 6 days after inoculation, which is kind of early. And at those stage, the nodules is still visible, and we want to trace like how they looks like in the early stage of the crack entry. And here are the acknowledgements. I would like to thanks to all my lab members for their help, and I would like to thanks to the funding source from the National Peanut Board. And thank you all for your attention, and I'm glad to take any questions.
Thank you very much for the presentation. We have time for questions. The first question that comes is, do you have any information or do you have any idea on the ecological competitiveness of these transformed bacteria, the Bradyrhizobium? If they were to be put back into the peanut fields, is there any information on how competitive they would be or compared to strains that are already being used? You'll need to turn your microphone back on. There, that's good.
Okay. So we haven't do a field test about this strain, but based on the previous study, there is a strain is also modified with the insertion of the plasmid, and they do it in the soybean, and they find this strain is not influenced by their like ecological compatibility. So I would guess this one is also not influenced.
Okay. A second question there is, do you have any idea if this is successful, how long it might be before, are there plans being developed for growers or for commercial use of a product like this or of a strain like this?
I think this GFP strain, it provides a tool to study the plant and microbe interaction. So we can use this one to trace the bacteria in the crack entry, or probably there are some other applications. But right now, I haven't think about like what kind of other applications this strain can be in the field.
Okay. All right. Thank you very much. That concludes the questions we have, and this is also concludes our final presentation of the session.